Hello, hello, hello. Welcome, everybody. Welcome to the stream, y'all.
Hello.
Today, we have a very special guest, very special journal club. But before we get into our special guest, as well as what we are doing today, I just want to say hi to all of you who have shown up in chat. Hello, Toxo. Hello, callsign Bread. Glad you could make it to the stream. Hello, Resta.
Hi, Resta.
Hello, Thomas the Thermonuclear Bomb. Hello. Welcome. Yeah, that's my boy, Blue Small Guy. That's me. I am the Blue Small Guy. And
I'm
today,
also a Blue Small Guy.
Exactly. So let's have our guest introduce himself, please. Guest, introduce yourself.
Hello, my name is Spollax. I am a clam VTuber. I tend to hang out more on Twitch. So this is actually the first time I think I'm on YouTube. I mean, it's a very novel experience for me. Thank you so much, Vi. Lag. No, I'm sure it's fine. But yeah, I tend to not do much science streams, but I am a scientist or some sort of derivative of. I tend to play a lot of roguelites, a lot of Nintendo games, a lot of fighting games. But I guess I'm branching out of my comfort zone a little bit with like an educational type stream, but definitely not outside of like my lifestyle, because I was literally just doing science earlier today. I took a short non-science break, and now I'm back to doing science.
Yeah, so I figured before we get into the paper, just to inform the audience that I didn't like drag a random.
please help me. He's kept me locked up in his basement.
Shh. Yeah. Yeah, just to inform chat that I haven't dragged like a random VTuber out to assault them with science. Can you tell us a little bit, or I guess I'll go first in terms of like reminding my audience in case they haven't heard. But yeah, I went to grad school for biology. I studied the immune system within neurons. So the nervous system is what we call immune privileged. But there are still immune proteins inside neurons, and they still do things, and they actually have functions that the normal immune system doesn't have. So they actually help with things like having neurons communicate with each other, and that's pretty cool. So this paper that I've chosen is actually a little outside of my comfort zone, but it is more in the realm of our guest here. So can you explain a little bit about what you do professionally?
Yes, I am a research assistant, aka research tech, aka also sometimes lab manager. I am fresh-ish, couple years out of college with a biology degree. And I guess to talk a little bit about my history, I had bright ideas or ideals and bright plans to be a doctor in college, as many people were. And then my senior year of college, I was like, this is kind of making me really unhappy. So I ended up pivoting over to research, which I had been doing at the time, working in microbiology labs, and decided to continue down that kind of path more professionally. my job entails basically around 70% is like bench work. I work almost strictly entirely in the wet lab, and then the other 30% is involved in like lab manager responsibilities. So hiring new people, training them, supply management, equipment management, all the little things that grad students kind of take for granted sometimes. Cough, cough. Just kidding. But making sure that the lab runs smoothly, and that I can help out where possible. And that also kind of applies to my research, where since I'm in microbiology, and if I was alluding to this, but I also do tend to work a lot with proteins, specifically small proteins, looking into their capabilities as either a new antimicrobial or trying to utilize the secretion system of these small proteins for anything, for like some sort of delivery, some sort of or maybe some sort of eradication of a hostile pathogenic bacteria. In terms of work, I guess, that I'm doing, so that's like kind of like broadly the projects that I'm usually working on. And in terms of like the work that I'm doing, it's a lot of like DNA work, cloning, protein expression, protein purification, DNA gels, protein gels, various like bacterial assays that are pitting bacteria against each other or against some sort of
Whoa, what
antimicrobial.
the heck? You do bacteria PvP?
Kind of, yeah, in a way. Usually it's pretty one-sided, unfortunately.
What the heck?
Um,
Wait, say more.
yeah, so
Who's the strongest bacteria in this
unfortunately,
PvP?
it's whoever we engineer to be stronger typically.
Oh, dang.
Sometimes, very rarely, are we working with like strictly wild type. Although sometimes we do that, to test their, um, the ability to colonize or outcompete different strains. But typically, we engineer bacteria, lab bacteria, sometimes various strains of lab bacteria like E. coli or
Hmm.
Acinetobacter baumannii or Klebsiella pneumoniae to secrete some sort of protein of interest that we want. And then we, either incubate it with a weak like target strain of bacteria, or we put them like on top of each other in like some sort of agar plate medium, some sort of solid medium as opposed to like a liquid medium, and then have them secrete whatever thing they're secreting in order to kill bacteria. And then you can
So,
kind of just like optically tell.
just to calm the fears of the audience, you mean to do this for the betterment of humanity, right?
Yes. This is all the betterment of humanity. I can get a little bit, well, I mean, this is kind of like I want to say like lab meeting pitch is that we always tend to use this like in the beginning of like lab meetings or beginning of presentations, is that not to like, make anybody too scared because it's not that bad, but some experts do consider us to be in a what is it called? A post-antibiotic age, where the rate of discovery is significantly lower than the rate of like antibiotic-resistant bacteria. But we are fighting the good fight, trying to discover new pathways or new, completely like novel antibiotics, which is very difficult, because a lot of the antibiotics we have, they tend to just be new variants of an existing class of antibiotic. So you got your like penicillins and your and in that one, you have like amoxicillin and some of these other carbanicillin, ampicillin, all these different variations of it, which, although useful and can be used in different applications, they unfortunately tend to follow the same mechanism of resistance. So we're trying to find wholly new things that bacteria haven't evolved to uh, resist yet, so that hopefully we can find something. Or if not, finding something directly from our lab, we can help other labs kind of, develop these assays or these screening techniques or these high throughput methods that they can instead use to also discover, their own neat little thing that they can use to hopefully, I don't know, maybe 10 to 15 years down the line, whenever, the what is it, the
Yeah, yeah.
FDA
You're
allows
at
us
a
to do stuff.
you're at the basic science level, right? You're not really at the
Yeah, we're we're
clinical
we're the
trial kind of level.
definitely we're on like the very first beginning parts
Yeah.
of the
So
research pipeline.
I think that's a really good transition for us to actually start talking about the paper. But before I get into that, I want to address the current poll. So we have more than half of the audience at a high school or less biology level, which is actually going to be very cool because Spollax and I have been talking about behind the scenes. This is a difficult paper.
Oh, this paper's real dense.
This paper is pretty tough. Okay, if I'm going to sing the praises of PLOS every day until I die, I love the fact that the journal is open source and I can pretty
Hmm.
much pull whatever and cover it on a stream. But sometimes they release papers that are very hard to wrap your mind around in terms of the narrative. That doesn't mean that you should click off. What that does mean is that if you are able to follow along, and I've tried my best to make this accessible, if you can follow along, I bet your IQ will like increase by 10 points just by sitting through the stream. Because I was messaging Spollax like all day yesterday being like, God dang, this paper is so difficult.
Yeah, we're kind of kicking ourselves a little bit for picking a paper like this, but
yeah. But I think it's cool. I think it's cool. It is a cool paper. It's just that the experience of reading the paper was quite challenging. So be warned. So to go off of what Spollax was framing, so we're interested in finding new ways of inhibiting or killing bacteria, because the more things that we find that kill bacteria and don't kill us, the better prepared we'll be when all of our antibiotics stop working. You don't want to think about that kind of future, but that's sort of the gist of why we are interested in learning more about molecules with antibacterial properties, right?
Correct.
The first stream I get a hard paper, grr. I'm really sorry.
Have fun, yay!
Have fun. And also, we'll get through it together. I believe in us, okay?
And also, not to scare people, but knock on wood, it'll never. This current antibiotic resistant bacteria situation won't actually like affect the general population. We're looking at it because it's our job to look at it. Someone's got to be looking at all this. We want to be
prepared for the future. Yeah.
Yes, definitely. Because if we ignored it, it could be, but we're not, and so it'll be fine.
Yeah. Trust. Okay, so let's get into the paper title. Oxidative stress-induced... Oh, I mistyped it. Oxidative stress-induced in E. coli by the human antimicrobial peptide LL37. So LL37 is the molecule of interest that we will be talking about in today's Journal Club, so please keep that in mind. That's the peptide that we're analyzing for its ability to destroy E. coli. Okay? It's published in 2017 in PLOS Pathogens, so it's a bit of an old-ish paper, but I picked it because it sounded cool. All right. Our bacteriophage still on the table. So if we're talking about alternate methods of bacteria control, it absolutely is. Maybe we'll talk about that one day.
Yes.
So a little bit about antimicrobial peptides. This is a class of molecule that we don't typically talk about very often. And in fact, on my channel, I kind of have a bias actually for the pathogen. If you watched any of my videos, you would see that, the bacteria are so tremendously clever, the viruses are very clever, the immune system fights back, but the bacteria, they always have something up their sleeve to counter us. That's not always true, however. Sometimes the immune system also has a pretty neat trick up their sleeve, and this neat trick that we're going to be talking about are antimicrobial peptides. So these are a vast array of different peptides that the immune system can make. Although now that I think about it, I don't know if LL37 is human derived, but some species makes this. And
I think it's human derived.
I think it's human derived too, but I wasn't entirely sure. If you have the paper open, you can check, but I actually don't. I just have my slides.
I have something better. I have Google.
Oh, very good. I will continue to talk on this slide then.
Yes, it's human derived.
Okay, great. So you make these short proteins, and these just wreck microbes. Like, I feel like you don't talk about the ways outside of immune cells in high school. Like in high school, I believe you learn about T cells and macrophages, at least a little bit, but you never really hear about antimicrobial peptides. So that's why I'm pretty excited to get into how these actually work. So antimicrobial peptides, or AMPs, can disrupt microbial intracellular processes. So not all antimicrobial peptides do this, but they can. They can do things like stop bacteria from making DNA, which means that they can't multiply. They can stop them from making RNA, which means they stop making protein. And they also can block key enzymes, which might interfere with the bacteria's normal way of life. However, there is something that is more common, and that is AMPs can tear holes into bacteria. And Spollax, you might be a little more familiar with this, but I'm going to get through a little bit of a biochemistry lesson before we can geek out about how antimicrobial peptides work. So let's take a look at this graph right here. this barrel, this red barrel, represents antimicrobial peptides. And there are actually a couple of ways where, if you know a little bit of chemistry, you can actually see they can actually tear holes. So if you imagine this as the bacterial cell wall, or I shouldn't say that, a bacterial membrane, you see that there are three principal ways by which these peptides can poke holes in that membrane. The antimicrobial peptide can just nudge, like create these gaps in the membrane. So this is the barrel stave model. There's the toroidal, toroidal, maybe, where it kind of pushes the membrane in and pokes holes that way. And then there's this one, which is my favorite one, this carpet-like structure, where the antimicrobial peptides can tear, like rip chunks off of that membrane. It's absolutely insane. Dang, that's gruesome. Yeah, exactly. So the fun thing about these models is that if you know a little bit about chemistry, you can actually visualize how these peptides do this kind of thing. So membrane busting may have mechanistic explanation rooted in biochemistry. So the lipid bilayer of a bacteria, located here, the phospholipid head, these circles here, these are all negatively charged. So phosphates have a whole bunch of electrons, and so that gives them this negative charge. But in the middle, these tails, these are called lipids, and they are nonpolar, so these are not charged. These here in the middle are afraid of water. Out here, they love water. And the thing about antimicrobial peptides is that you can actually physically understand how they break cells. If you have an antimicrobial peptide that has positive charges on the ends, then you can see that these positive charges will be attracted to the negative heads, and then that explains why this can happen, this like pore can form. And if the amino acids in the middle are non-charged, then they can be happy with the tails, and then everything will be fine. So the thing about this tail is that it really doesn't want to touch water, and if you poke that hole, you will have the lipids in contact with water, which they don't like. So these antimicrobial peptides can fill in the gap. Similarly, other antimicrobial peptides might have different charge properties where if you have enough positive charges on this side, it can interact with the negative heads and keep it from forming one continuous layer. And of course, here in the carpet-like system, you also have a bunch of positive charges that like binding those negative charges. So hopefully you'll understand a little bit about the mechanism by which these antimicrobial peptides work. It's very, very, very cool. Spollax, do you have anything to say about antimicrobial peptides? Because that's where my understanding ends.
Yeah. So the reason I Phi gave me a couple of papers and I was like, I want to do this one is because although I don't specifically work with antimicrobial peptides, there are a couple people in my lab who work pretty heavily with antimicrobial peptides. And so I literally saw LL37 and I was like, oh, hey, I know that one. I've heard about that one before. And I read it a bit more. And so I wanted to read this paper. But yeah, that's a great explanation of how it works on like a more biochemical aspect. I tend to kind of shy away from biochemistry. Although I do know, like I do understand like the more basic processes. It is something that's a little bit spooky. But yeah, bacteria, most living things, I should probably say, really like to kind of be intact and have all of their insides on their insides and have all of the outsides on the outsides. And so whenever you go ahead and do this, which is called like membrane permeabilization, because it's causing the membrane to become permeable to various different substances. Usually these substances are very specific and like kind of tightly regulated by what can go in and outside of the cell. But whenever you start punching big holes in it with like an antimicrobial peptide, for example, it'll cause stuff to leak out. It'll cause stuff to leak in. It'll also disrupt some sort of, typically a cell will work really, really hard to maintain kind of a charge balance or charge imbalance, I should say, where it wants to keep part of the cell one way and wants to keep outside of the cell another way. And whenever you disrupt it, then, things start getting weird and then the cell actually isn't able to function. I wish I had a bit more to say about that. There are some more specific things. Maybe you have some slides prepared about that, so I don't want to step on your toes, but.
Yeah. So the other thing that I want to say is that membrane permeabilization is the official way to call this. It's a hard word, so I'm actually just going to call it membrane busting.
Yes.
I think membrane busting is cooler too because it reminds me of StarCraft 2. I don't know if you've played StarCraft 2, but there's
I have
this
not, unfortunately.
rush strategy called Baneling Bust, where you would make a unit that would destroy the enemy's defenses at a certain time. So I find that that's a little more evocative of
Yeah, it's
power.
quite akin.
All right. Okay. So let's go to the next slide. Oh, okay. So this is a absolutely sick picture that we're about to see. So this is I believe it's scanning electron microscopy. This is an actual picture of E. coli. It's been pseudo-colored. So, I mean, E. coli aren't actually orange, but it's been colored for your convenience. So this is the before picture. Okay. This is before we add the AMP. And I don't know if you've seen this picture, Spollax. This is nuts.
I don't think I have.
You add the AMP and oh, look at that.
Oh, sorry, I'm on a little bit of delay. Ooh.
Look at that. Oh, do you know what that green is?
I do not.
That's the bacterial chromosome.
Ooh.
Yeah, that's just the DNA leaking out. Yeah, Cheeto guts, exactly. That's crazy.
They typically like to keep the insides on the inside, and when they're not, it's usually not a great time.
I mean, that is brutal. I feel bad for the E. coli. Like, it's hard to sympathize with the Cheeto, but
they deserve it.
You have commensal E. coli too. I wouldn't paint them all under the same brush. You don't know. Okay, but yeah, so AMP's super cool molecules. Their ability to bust membranes make them pretty interesting targets for research in a post-apocalyptic future where we don't have our antibiotics working anymore. So so better to get on that research now rather than later. Yeah, bro got busted, exactly. So you might be wondering, can AMPs be made into therapeutics? And the answer is absolutely. In fact, vancomycin, which is something that a couple of you may have had if you've had a particularly severe infection, is an AMP from the soil bacteria. I'm not even going to try to pronounce that.
Amycoleptosis orientalis. I think.
Dang.
I don't know. I've never worked with it before, so I could be wrong, but I'm just saying it with a lot of gumption, so you think I'm right.
You have more gumption than I do. Oh man, yeah. So vancomycin is one of these AMPs, so it's actually of great research interest to try to find more of these, just in case our current antibiotics fail.
I had no idea. That's cool.
Yeah, I didn't know either. Aren't I a neutrophil? E. coli can still be friends to neutrophils. You know, we have a mutual agreement in the gut, which, by the way, after I finish the Prion video, stick around for a gut microbiome immune system showdown video, which is
that'll be very fun.
Going to be very spicy.
It's all good whenever they stay inside where they belong, and if they start leaking into like your bloodstream and stuff, that's a little bit less good.
Exactly,
from my understanding.
When they are in the wrong place at the wrong time, that's when we get kind of mad. But if they stay where they're supposed to stay, we're all good. We're friends even. Okay, so we're interested in learning more about these AMPs. And by the way, this image right here is the AlphaFold. So
AlphaFold.
Yeah, AlphaFold is Google DeepMind's AI protein folding program where it can predict, it doesn't confirm, but it can predict what proteins look like if you can't crystallize them. So this is what we think LL37 looks like. And so this is our representative AMP. We know that it can do things like inhibit DNA, RNA, protein synthesis. It can inhibit enzymes. We know it can do membrane busting. But it's also worth investigating what else they can do. So maybe there's some other mechanism, and we're going to be trying to figure out what LL37 actually does. By call sign Bread, thank you for tuning in. So here's a research question. What is the bactericidal mechanism of the antimicrobial peptide, LL37? The reason why we care is that learning more about this particular peptide can help us optimize this peptide for usage in humans eventually down the line. So that's kind of the headspace that we're in. We're trying to investigate this peptide without too many directed hypotheses. Okay, so what's one potential way that any antimicrobial could work? Well, there's this thing called oxidative stress. So never underestimate the destructive power of oxygen. Oxygen is extremely deadly, incredibly reactive. And the thing is that the reason why we don't just evaporate and dissolve into dust is because our mitochondria protect us from oxygen by making it into water. And in the process, we get out energy. You know, very impressive. That is a powerhouse of the cell for you. It can turn this deadly molecule into energy and water, which is very cool.
Mm-hmm.
If you screw up this process, you can generate these things called reactive oxygen species, or ROSs. On the left here is an example. It's called superoxide. It's your normal O2, which you can breathe and be fine, but it's got an extra electron on it. So if you screw this process up in the mitochondria, you can generate these super reactive oxygens with extra electrons, and that would be very bad for you because these molecules can destroy your enzymes. It can destroy proteins. It's bad news. And this is also why you get advertised, blueberries and stuff like that, because they have antioxidant properties. They have antioxidant properties because they can help you manage the levels of dangerous oxygen in your blood. Toxo says, stop breathing, okay, TY. I am not a doctor, so I can neither confirm nor deny, but I would... my general stance is that continuing breathing is not harmful. Okay, so this can cause us to ask a couple of questions. Do AMPs, including LL37, cause oxidative stress in bacteria? We know they do the membrane busting thing. We know that they can inhibit all sorts of enzymes, but do they also screw with oxidative stress? And does this peptide in particular require oxygen to work? So by understanding these parameters, we might be better able to optimize technologies surrounding these peptides in the future. So this is the first figure in the paper, and I want to give you all a little bit of time to look through this data. First, I'm going to just give it to you normally, without any hints, including for you, Spollax, in terms of, I don't know how carefully you looked at every figure, but we can remind ourselves what this figure looks like. And I just want to let the chat kind of peruse this table to see if they find anything interesting. And if you find something interesting, you should let me know in chat. So I'll continue to vamp as chat kind of looks at this table. And don't worry if this all looks like gobbledygook. It's the exercise. Even if you don't understand what's going on, if you try, that's still exercising your brain in a certain way. So the thing to note here is that the growth, I guess I'll go column by column, right? The strain, so WT stands for wild type. This is your run-of-the-mill, not mutated in any special way, typical quote-unquote bacteria. And then you have a bunch of genotypes in the middle that are slightly different, and you can see how they're different here. And I'll say that if you don't have a biology background, you can pretty much ignore everything that doesn't have a WT here, because these are very specific hypotheses that don't really matter for our discussion right now. The growth condition here ranges from aerobic to anaerobic, and then we have anaerobic plus this molecule, which I'll get into in a little bit. Aerobic means that it is grown in conditions that contain oxygen, and then anaerobic is without oxygen. Doubling time, so E. coli reproduce very, very quickly, so it takes 50 minutes for, roughly, in this sort of state when you add the peptide. So after, because correct me if I'm wrong, Spollax, but I think uninhibited, very happy E. coli, their doubling time is like 15 minutes.
I was gonna. The one I've always heard is that the doubling time of E. coli is usually around half an hour.
Oh,
but
half an hour.
That might be for our specific lab strain that we tend to work with. Lab strain being just like a weak, wimpy strain of E. coli that's not dangerous, that's suitable for lab work.
Can I ask what strain? I'm just kind of curious.
Typically the one we use is DH5 alpha. It's a very common cloning strain. It's got a couple of modifications that make it easier to clone into. It won't destroy its own DNA as easily.
Yeah, DH5 alpha is very common. I've used DH5 alpha before too. Yeah. I'm interested to know what makes it weak, although that's maybe a conversation for something else, for some other time. And then MIC, this is minimum inhibitor concentration, and in parentheses it's got micromolar, so if you haven't taken chemistry yet, then this is maybe not meaningful to you, but it's a measure of concentration. So the higher number this is, the more concentration we have. I'm concentrating on concentration and it's not working. You may want to check if you have been hit for damage, and you may want to check if you have passed your Constitution saving throw. So Resta's got a comment. E. coli grown under anaerobic condition have a higher MIC. That is correct. Very good, Resta. So this is what my eyes were drawn to, right? So between the wild type, anaerobic and aerobic, we have an increase, almost fourfold concentration of the peptide that is required to stop it from doubling. So I think that's pretty interesting. So I'll go ahead and say that that is the thing that I noted. And so we can conclude that greater AMP concentration, so more LL37 is needed to inhibit E. coli cell growth without oxygen. In other words, it works better with oxygen and it's kind of crappy without oxygen.
Mm-hmm.
So that's kind of interesting. What about this last column, this last row? So they added this molecule. I wish I looked up this beforehand, what this molecule is called, KNO3. I'm sure it has a name
Potassium nitrate.
Yeah, potassium nitrate sounds correct. All the chemists are face palming right now.
This is potassium nitrate.
Thank you. So potassium nitrate enables anaerobic respiration but doesn't introduce oxygen. So basically what this means is that, so E. coli, they could do with oxygen, they could do it without oxygen. And adding KNO3 allows you to access anaerobic respiration so that you don't starve without oxygen. Because when you change up the growth condition, and Spollax can correct me if I'm wrong on any of this because I'm less familiar with respiration in bacteria. But basically, there's more than one thing changing besides just oxygen levels, right? Like, it's not that surprising, maybe. Actually, I think I'm going to walk that back. I think I'm just going to leave it there and maybe come back to that idea in a future slide. Basically,
sure,
it works better with oxygen than it doesn't. So that kind of indicates that maybe oxygen plus LL37, maybe they interact somehow to hurt bacteria. Okay, so let's continue with that in mind. So we know that it needs oxygen to work the best, but does it cause oxidative stress? So that's actually a different question that we would need a different experiment for.
Mm-hmm.
And you might think that this is an obvious question because oxygen is required for oxidative stress. You heard me earlier in the lecture say, or I should say journal club, this is not a lecture. Oxygen is required for oxidative stress. So you would think that if LL37 needs oxygen, which is required for oxidative stress, then that would naturally mean that LL37 causes oxidative stress. Does that make sense? That's a tempting line of logic. But I want to challenge chat. Why are we not done with the paper already? Our goal was to ask whether or not LL37 interferes with oxidative causes oxidative stress. Why are we not able to just end the discussion right here and say, yep, that's what happens? Why? And so this is what I was saying with, like, if you are a high schooler below biology, like, you don't need biology education to figure this out, but it does help. This is more like a logic puzzle for those of you in chat who want to challenge themselves. I think it's interesting just because, like, when I was reading this paper, I was struggling a lot with sort of the logic between the sets. Like, I found myself pondering this as I was reading the paper. I was like, wait, didn't we already show that LL37 causes oxidative stress because it needs oxygen? So, chat, why are we still here? I'll give you a little time to think about it. And Spollax, I'll wait a little bit of time, and then maybe you can offer some of your thoughts in terms of why we're still talking about this paper.
Sure.
A little bit of
There's no wrong answers.
Well, there are wrong answers.
Well, what did I want to say?
I'm talking directly to you. Chat, there are no wrong answers.
For chat, no wrong answers. For me, there are wrong answers.
For you, there are wrong answers. Yeah.
Oh no, I have expectations.
Yeah, chat, if you just come up with anything, any answer, if you attempt, that works out your brain, you've got your brain workout in for the day, even if you're wrong. But if Spollax is wrong, then I will... No, I'm joking, of course. Maybe the kill mechanism uses oxygen as a reactant, but it's not oxidative stress. That's a very good hypothesis, Hairday. Very good. Zeal, LL37 isn't an oxidizing agent. Maybe it's more a catalyst to encourage O2 to interact with whatever. Also a great hypothesis. Fantastic responses. Spollax, what do you think?
Uh, so I did well cheat a little bit, I guess. I phoned a friend where I brought up this paper and I sent it to a couple of my lab mates who actually do work with antimicrobial peptides, and they've actually I think worked with LL37. And I kind of polled and got their opinions for it. And one thing that they mentioned to me that kind of like reframed how I was viewing the paper was that um oxidative stress isn't it's a very broad kind of like symptom of a cell's like dysbiosis or like being unhealthy, I should say. Um, it's kind of a chicken and egg thing. Like, does it cause oxidative? Does the fact that LL37 kill better under aerobic conditions mean that it's causing oxidative stress, or is some sort of oxidative stress that's already happening allow LL37 to operate better? And I guess that's kind of like the issue that, or like the question that they're trying to figure out.
Exactly. I love those answers. So I'm actually going to make Hairday and Zeal's answers a bit more broad because you have specific hypotheses, but I'm going to generalize them so I can remain more agnostic, right? Those are very testable hypotheses, but I want to remain as tabula rasa, blank-minded as possible. So I'm actually going to say maybe there's some other hypothesis, some other metabolism thing gets screwed up through LL37 plus oxygen, which is exactly what Hairday and Zeal have suggested. Like maybe that's not what LL37 is doing, and the oxidative stress could be coincidental or not happening at all. So that is why we cannot stop at this point. That there is more to be learned, but I love those answers. That's really, really great.
This is, oh, if I
Yeah,
may, this is like a prime example of, correlate. You always hear correlation does not equal causation, and this is like a prime number one example. Well, maybe not number one,
exactly.
but because it's
No, I
not the
think it's
easiest thing
great.
to understand. But there's something I wanted to say, and then I forgot what I was gonna say.
I'll vamp for a bit longer if you need to.
Yes, hopefully I can try to remember. Wait, no, I don't have it. Well, maybe it'll come back to me later, and I'll just interrupt you
Yeah.
then.
So I'll say this: like, you hear correlation is not causation all the time, but it's actually really hard to always be aware that there is that disconnect, right? Like, that's why I love teaching biology so much. Is because you get so many instances where you have to be very careful about your own logic. It's funny. I was actually talking to Cideray earlier about how complicated psychology is, and I feel like
hmm
it's like it works out like different muscles because while I was discussing some research with her, there was so much complexity in those systems, so many like confusing correlates and factors that are very just messy. Like when you ask
hmm
a molecular biologist, there's this idea of messiness, and I think psychology is very messy, but sometimes for certain people that can be a lot of fun. I think for at this level of molecular biology, it's tight. It's messier than, of course, physics is, right? But in terms of
Right.
the logic lines, like this is the kind of workout that your brain gets when you do molecular biology. It's also messy when you get too broad, but in terms of you're just talking about one molecule, this is the line of thinking you need to be doing.
I remember what I was gonna say.
Nice.
Woohoo! And this is more of just like a broad like microbiology research thing. Is that broadly, that my word is not gospel. I'm not the most qualified person in this field. That's definitely to be said. But it's very easy to kind of show that something impacts a cell. This thing causes this. Having more oxygen causes the MIC to be lower. It's very easy to kind of prove that. It's much, much, much more difficult in order to prove the specific mechanisms and why things are happening and how things are happening. And that's why these papers tend to be very long and very verbose and very like specific, is because you can't really, well, I mean, kind of, you can't really build a story or like get up the paper published by just saying that like oxygen does this. The committee that's going to review the papers can be like, we need something a bit more substantial. But once you start getting into the weeds, then that's when things get very complicated.
Exactly. And Alexis, you are spoiling the end of the paper. We will get to that. Okay, so let's move on because Alexis is spoiling things.
No spoilers, please.
No
No.
spoilers. Although you can read the paper, I have it pinned, but no spoilers.
hmm
Okay, so we want to link LL37 or not link LL37 to oxidative stress. This is the hypothesis that we want to test. The eventual goal is to see whether or not this peptide does something to oxidative stress, right? Okay. If I knew what that meant, I would be surprised. We'll get to it. We'll get to it. Biology is a lot of vocabulary. That much is true. Okay, so what does LL37 do to E. coli exactly? We know it's an AMP, so we have some reasonable hypotheses that it can bust membranes like our other antimicrobial peptides. But it's time to take a little bit of a closer look at E. coli in general. So E. coli is a gram-negative bacteria, which means it's got all these layers. And I want to give chat a little bit of time to think about how you would molecularly test what LL37 does to membranes in E. coli. I would be shocked if anybody gets this answer correct, but just as an exercise, right? Just as pushing your brain to think about attacking a scientific problem. And if you don't have biology education, it's totally fine. You can just guess, and I'm not going to penalize you if the technology that you want to do, it doesn't exist, right? So, be very creative, and then I can tell you if I know of a technology that fills that kind of role. So I'll vamp a little while chat comes up with a way to test. And again, I don't think anybody would figure it out. So there's really no expectation that you get it right. Because when I learned how they did it, I was like, never in 1,000 years would I have, I mean, maybe in 1,000 years. I don't think in a month I could have figured out what they actually did. Setting us up for failure, not cool streamer. My bad. You know what? I should reframe it. If anybody guesses what they did, I don't know, I'll gift a sub after stream or something.
Yay!
Could you modify bacteria so they only have certain layers and see what happens? Very interesting hypothesis. I don't know, Spollax. I don't know if you can do that.
I hate to be the bearer of bad news, but probably not. Removing all of these, well, periplasmic space isn't really a layer, kind of a layer, but like the inner membrane, the outer membrane, and the peptidoglycan layer are all very crucial. If you try and remove one of them, the cell will just like kind of die, or it won't be able to like reproduce.
Yeah. So
what is a high C experiment?
I don't know what a high C experiment is.
Like the drink?
Otto, have a dye which can't penetrate the membrane normally that attaches to DNA. Ooh, okay. Yeah. Okay. I think I have to gift the sub, Spollax.
Basically, yeah.
Can I do that right now? I don't think I entered my information to gift a sub exactly right now. Sorry.
Hit the nail right on the head, though.
Yeah. Dang, very, very good.
Microbial student. That's why.
Half credit. You don't get full credit if you're a microbial student and you come up with that.
That's cheating.
That's cheating. That's cheating. I don't think I can gift a sub right now, but I will gift.
No.
I'll gift one sub for that answer. I'll gift one sub for that answer later. Okay, but yes. So the reason why I give you time to think is because when you see what the scientists actually do, it takes time, right? If you try to attack the problem and then you see what the scientists do, there's this effect of like, damn, that's clever. But Otto, you got half of the story. So the authors use a very special bacteria that they engineered to express a protein called GFP. You pulled us. It's your fault for not thinking the microbial students aren't out to play. You're right. Scam. Okay, I'll gift 5. I'll gift 5. When E. coli make GFP, they transport that GFP to what's called the periplasmic space. So it's kind of hard to see with these arrows, but this teal layer, that's where the GFP gets stored. And so when the outer membrane is bust, that GFP is going to leak out into the open. So when you see the bacteria, if it's healthy, it'll be green. And when the outer membrane is bust, that GFP will leak out into the environment and it will stop being green. And so this is the part that Otto Liu is referring to, not only did they use an engineered bacteria that expresses GFP, they also added a dye called Cytox TM, green nucleic acid stain. Not a sponsor, although I am open to any and all biotech sponsors if they
Man,
ever.
if we could get sponsored by Thermo, that'd be
Oh,
crazy.
I would love Thermo or
Thermo
what are their
sponsor us.
competitors?
New,
Sigma Aldrich,
New England Biolabs,
New England Biolabs. You know, any one of you wants to sponsor me, my DMs are open.
Fisher. Do we say Fisher?
Are Fisher and
Fisher
Thermo
also.
Fisher different?
I
I always lump them in the same.
I think they're. I don't know. Fisher Scientific versus Thermo Fisher. I'm not sure.
Anyway, anybody.
Moving on.
DM me. Yeah, moving on. So the thing is that only detects whether or not the outer membrane was compromised. They used a dye that cannot penetrate the bacteria normally, but when it does penetrate into that bacteria, it binds that DNA, and only when it binds DNA does it fluoresce green. So we are looking at two different signals here. We're looking for an initial disappearance of green, which would indicate that the outer membrane of E. coli was breached. And then we're looking for the reappearance of green. And it drives me crazy, Spollax, because they use green and green. So when I was looking at their data, I was like, what the hell am I? I don't understand. It's just, okay, we'll look at the video. This is a very exciting video.
Mm-hmm.
So this is time-lapse footage of LL37 breaching the E. coli membrane. Very cool. I love doing these streams because I love showing what actual real data looks like. So on the left here, we have something called a phase contrast photo, phase contrast time-lapse. And what this basically is just, you can imagine just like a camera, like a normal light camera. Although I didn't read up on how phase actually works, but think of it like a normal light camera. And then on the right, we have fluorescence. So this is the green signal that we are looking out for. So again, when we see the loss of green, that is the green fluorescence protein leaking out of the outer membrane. And the resurgence of green is the Cytox stain binding the DNA. Okay, and then we'll watch this video. We'll loop it a couple times. So we see that green after the protein has been added. And that's, oh, it gets so bright after that Cytox gets in.
Getting flashbanged.
Yeah, exactly. It's basically getting flashbanged. So you'll see that there's a number down here. That's minutes since LL37 was added. So the video, I think, starts at 0 minutes. So right when that peptide is added.
I think it starts at like -5. I think,
yeah.
Just
okay,
as like a control.
Yeah. Okay, so when it's added, that membrane is bust, and then the Cytox comes in, and it's really bright. So if we look at it, in this particular E. coli, this happens really fast. We'll watch it one more time.
Yeah.
So
that's pretty nutty.
It, and then it fades like 6-7 minutes, and then 5 minutes after, it's blazing bright with the Cytox stain. That's so crazy. I love this video. It's so cool. This was much more of a visible reaction than I expected, honestly. This Cytox stain is so bright.
Yeah. What is this? What is the word I'm looking for? Like
the gain
imaging, I guess. Just like being able to see stuff, see the bacteria you're working on is very, very cool.
Yeah. So
the individual cells.
Yeah, I love microbial where you can actually visualize cells. It's neat. So this is the time lapse photos. So not the actual video, but a Green Lantern E. coli. Exactly. It's crazy. And you'll see that at 5 minutes, this is when the fading starts. By 8 minutes, almost all of that GFP is leaked out of the membrane. And then like a minute later, that Cytox stain is making its way back in, which is crazy. And you'll actually notice that the GFP is not very bright, so they had to turn up the gain 10 times to get it like this. But then they had to turn down the Cytox stain because it's so bright. So again, yeah, sorry, Cytox TM. Yeah. So the first green loss, again, is outer membrane breached. Green gain is inner membrane breached. And if we plot this with the cell length, so as E. coli grow, they get longer. And if they are, if they're hurting, then they shrink. They like shrivel up when they are damaged, at least as far as I understand. And you can see that these two lines temporally correlate. That when that outer membrane is breached at the 5 minute time, you see that's the high, that's like the happiest E. coli, they're growing, they're elongating. The second that outer membrane is breached, you start getting shrinkage of that E. coli cell. And that's when this, it starts shrinking, and then you get the inner membrane breach, and it just continues to crater. So this is about, what, like a 18-ish 16, 18-ish% decrease in length is pretty significant.
I just realized I've been reading this figure wrong this entire time. These things happen as a scientist.
Yeah.
Whoops.
Whoops. Yeah, so OMP represents the second that the outer membrane is breached. The CMP represents the second that the inner membrane is breached, which is why this shrinks a little and then why this shoots up like crazy.
Mm.
Very, very cool. Very, very cool. Okay.
I will say, when I read this the first time, I printed it out in black and white, and so I couldn't really tell. So that's why whenever I think you asked me a question about this, I was answering something differently. But I'm now realizing where the confusion was coming from. Whoops.
Gotcha. So this paper, again, was really difficult. Okay. The other thing, because they are testing whether or not LL37 causes oxidative stress, they wanted to check whether or not it worked better when there's oxygen around. So there are these 3 lines. Black represents aerobic conditions, so oxygen containing. Green is anaerobic, with the additional molecule that allows the E. coli to do energy normally, basically. And then fermentation, so this is conditions, I don't actually know. Oh, I think so fermentation is one pathway by which bacteria that don't have oxygen can make energy. And then KNO, I think they add so that they allow that bacteria to make energy mostly normally, is what I'm getting out of that. And what we'll see is that this acts as this fraction of cell membrane permeabilization. So at 0, that's like no bacteria have had their inner membrane breached. And at 1, that's 100% of the bacteria have had their inner membrane breached. And we'll see if we just draw a line at 30 minutes that with oxygen, you actually get more bacteria with membrane breaches, which is kind of interesting because that connection is not exactly obvious. Like, why would this peptide need oxygen to bust membranes? It's kind of bizarre, kind of an interesting to chew on, I think. You have anything else to say on this slide, or? That's where my thought ended, by the way. That's why I asked.
Nothing. Nothing. I don't think you already covered. CMP also just stands for cytoplasmic membrane permeabilization.
Gotcha.
I think it's useful to mention all the units and whatnot.
Yeah, it's biology acronyms everywhere.
I had to look. Don't worry. I'm looking at the paper on my other monitor, and I had to go read the description to make sure to check what the units actually meant.
That is fair. So we've generated 2 pieces of data that allow us to generate a novel hypothesis. We have the piece of data that says the peptide causes E. coli growth to halt, and we also see that it works better with oxygen, so we have a legitimate hypothesis now. We think LL37 halts E. coli growth through oxidative stress. It needs oxygen. Now let's think about how we can prove that oxidative stress is happening. So here I'm going to turn it back to chat. So again, you don't have to have biology experience. And Otto, I am looking at you. Don't answer right away. Let the entirety of the cellmates at least think about it before they're spoiled. Again, don't limit yourself to what you think is possible. Think creatively. Think abstractly. If you had access to any kind of magical technology, any like way to read or measure things, how might you try, and there are multiple correct answers here, how might you try to address this hypothesis? Kind of an interesting question. If your hypothesis is that this halts E. coli through oxidative stress, what might be a crucial piece of information that you might need to confirm before you're confident that you're right? Kind of interesting. This part I actually didn't really know too much. Oh, welcome back, Call Sign. You came at a good time because
yes, there's a question.
There's a question here. So, Call Sign, just as a recap, and as well for anyone else who's recently entered, there's this antimicrobial peptide called LL37. It hurts E. coli. We don't know exactly how it hurts E. coli, but we know it destroys or it doesn't destroy their membrane, but it pops a lot of holes in their membranes like Swiss cheese. And we have this additional idea that it causes oxidative stress, which is the same kind of stress that your body naturally generates that you eat blueberries to get rid of. You don't have to worry about the graphs, Call Sign. It's more the idea of the graphs. Could you test it by giving the bacteria antioxidants along with the LL37? Ooh. Interesting. That's genuinely an idea that I did not think of that would be very cool to see. I like that idea a lot, Hair Day. Unfortunately, They kind of do that later, but not quite, as we'll see. That's a great idea, and they sort of do that, but not quite. But I love where your head is at right now. So I actually like that more than what they end up doing. so at least you've won my sort of. Well, maybe they don't do it because it's not possible, which is another consideration to have. Add extra reactive oxygen species to some instead of normal oxygen. That is also interesting. So to see whether or not if you add more oxidative stress, does LL37 work better? Mm-hmm. That's a very cool idea. And so I want to think about whether or not that can generate useful data. And so I kind of want to maybe put Spollax on the spot. And
sure.
Do you kind of know what I'm hinting at? Why Thomas the thermonuclear bomb? That's an interesting experiment. I don't know whether or not it would generate great data. And Spollax, do you kind of want to offer an idea why?
Yeah, well, we'll see if we're on the same wavelength or not. But whenever you dramatically alter the environment of the bacteria, you kind of you introduce a lot of different variables and a lot of different changes that will make it harder to discern kind of like what the exact reason for it is. It'll be more difficult to say specifically whether or not it's LL37 that's causing the the difference, or if it's just maybe that amount of oxygen is poisonous to bacteria and that'll cause it to just be survive a little bit worse, a little bit less good.
Exactly. So yeah, that's 100% the same wavelength that I'm on. So don't throw that idea away, Thomas, because that kind of thinking can work for other systems, but we know that oxygen hurts, so that's not necessarily good. Use tagged
I did really
oxygen.
Like that idea, though.
Yeah, I really like that idea too. It might not be appropriate for this particular system. Toxis says use tagged O2 to see if those particular molecules become ROSs.
Ooh,
that's also really cool. Like heavy oxygen, that's neat. I like all of your ideas more than the actual paper. This is so frustrating. You've all come up with ideas that are cool, but that's not what they did. Okay.
Radioactive labeling is very cool.
Otto, if you have an idea, I'll take your answer too. At this point, we've generated some really smart ideas in chat, which is awesome, just awesome. Marking the oxygen could help. That's a little. I think Toxo and Alexis are around what they actually did, but not quite. Not using tagged oxygen, but the idea is there, Toxo.
Yeah. Now we're putting Otto on the spot.
Yeah. Now we're putting Otto on the spot. We're not graded. I'm not gonna give you a grade. It's just if you have an idea, I'll take it. But
we won't be mad, just disappointed. Kidding.
Yeah, only I get to be disappointed in Spollax because I've dragged him onto this collab.
Yes, I have stakes.
Okay. I'm gonna guess some way to quantify ROS. The microbial student does it again. Okay. Yes, to quantify reactive oxygen stress. Very good. But again, all of your other ideas, those were excellent ideas that are actually better at addressing this hypothesis. But that's not what they went for, and they have a good reason why they don't go for that, actually. But this is me trying to tell their story, and I'm not going to be perfect at it because I think the story that they told is actually really hard to follow. To measure oxidative stress, we get another proprietary technology called CellRox TM. CellRox Green. So CellRox Green is very similar to Cytox Green, which is the dye that we just used. It works very similarly. So what this does is that CellRox Green, I actually reversed this, but whatever. CellRox Green, we don't know, it's company secret. We don't know what molecule this is, but it reacts with superoxides or the hydroxyl radical. So these are two molecules that are an endpoint of oxidative stress. So when you have oxidative stress, you generate these superoxides. And so these superoxides can then damage this CellRox reagent. This CellRox reagent can then bind DNA, and then you get CellRox green fluorescence. CellRox fire. Yes, exactly.
Yeah.
I love cells rocks. So this is a measurement for oxidative. I always want to say oxidative phosphorylation because that term is just so burned in my brain at this point. It's a measure of oxidative stress. So Otto, you were right on the money. We're measuring oxidative stress, and Toxo and Alexis, you were hovering around the correct idea.
Mm-hmm.
Okay, so what does that look like? Oh, it's so cool, dude! I love that they have videos here. So let's take a watch here. Let's see. So again, when this dye binds DNA and oxidative, I always say, and superoxide, we get green. So we're at -5 minutes. So at 0, we'll add the peptide. So adding the peptide, 3 minutes, it's bright. In 3 minutes, it's this huge burst, and then it goes away. Isn't that cool? Whoa, that's kind of weird, right? It goes away.
I'm on a little bit of a delay. So unfortunately, you get 2 reactions then out of us. So
that's
more
true.
fun that way.
That's true. I forgot that chat can't see me watch the video live because I have a bit of a delay. So I'll play this again in a repeat and notice qualitatively what's happening. We get that surge of green at around the 3-minute mark, and then at the 13-minute mark it goes away. And that's really interesting. This is one of the reasons why the paper was so hard for me to read, because I was like, wait, did the oxidative stress go away? What happened here? So let's see. Chat, do you have any idea why the signal goes down? I agonized over this. I messaged Spollax yesterday and I was like, I don't know why this is happening. Is the cell dead after 13 minutes? Hair Day, they didn't test for cell death. Unfortunately, I cannot answer that. I can say that the E. coli is very hurt. That's for sure. Whether or not it is dead, I do not know. They did not measure that. It's a good hypothesis. You're actually hovering around the right idea. Channels. Okay, Alexis, say more. Or does anybody want to elaborate on what you mean by channels? Man, some of you chatters are smarter than me.
like this YouTube channel?
Like this YouTube channel? Yeah. True.
What kind of channel?
What kind of channel? What are you? I like that word that you're using, but I want to hear more.
Does this hurt the cell? I don't think they can feel pain, so no. Tentatively, no.
Yeah, so continuing to watch, if we combine Hair Day and Alexis's ideas, membrane channels. Okay. I think I can kind of tell where your brain is going. So the thing is, we have this cell, and we add the antimicrobial peptide, and we get this burst of oxidative stress. But we also learned that LL37 destroys or damages the inner and outer membrane. So Spollax kind of teased this early on in the talk, but when you punch a bunch of holes in the cell, stuff can leak out. So I was agonizing about this yesterday with Spollax. I was like, why does this green go away? And then I was like, oh, it's because we just poked a ton of holes in the E. coli. And actually, if you see, pay attention, chat, to the 12-minute mark, this left-hand side, it goes from opaque black to a little bit see-through. Isn't that interesting? I don't know whether or not that indicates that the E. coli has burst, because I'm not an E. coli expert. But the fact that when we look at the phase contrast, it goes from this opaque black color to going a little see-through just at the exact moment that dye goes away, kind of indicates to me that the reason why we get a decrease in green is that it's just like the cell just exploded, which is very cool. I like that a lot. It's neat to be able to deduce this kind of logic from the data.
Yeah, imaging is so cool.
Love that. So we talked a little bit about this. So LL37 does cause oxidative stress. It does cause oxidative stress because this dye that we add does measure oxidative species and measure superoxide. So then the follow-up question is, how does this peptide cause superoxide to be made? So this actually goes a little bit into how I figured it out from reading the paper. Saying what is oxidative stress? So as a natural byproduct of you just existing, your body makes these things called reactive oxygen species. O2, the thing that you breathe, very good for you, very healthy. O2 with an extra electron, very bad for you, destroys your proteins, destroys your enzymes. That's why you eat things like blueberries to clean up your body of these dangerous oxygen species. They're called antioxidants. So we want to know whether or not this peptide causes a buildup of oxygen species that can actually hurt you. So we'll see that the decrease in cell rock signal is temporally correlated. So in time, it happens. It has a correlative relationship with inner membrane permeabilization. So at the second that you start getting a decrease in cell membrane length, which is also the same time that you breach the inner membrane, you get this huge spike of cell rock signal. Although it's a little delayed, I would say, but it's around the same time. So that is indicative that that green going away could be just the cell leaking its guts everywhere. AKA cell broke, Lamau.
Cell machine broke.
Yeah, cell machine broke. Yeah, that's kind of what that is. What's frustrating is that they don't just say it. They're very coy in the paper. They're just like, oh yeah, cell rock signal decrease is temporally correlated with inner membrane permeabilization. And I was like, what? What does that mean? I don't really know. But very smart chatters in chat have figured out that this has something to do with holes being poked in the cell. Okay. So the thing that's difficult to tell about this story, or difficult to sort of... The reason why this paper is hard for me is because the storytelling is a little difficult. It seems that they kind of do some random exploratory stuff without really linking it together. But the idea here is that they want to get as much of a basic idea of how this is happening so that they can generate a plausible hypothesis. So what they want to do is they want to create a timeline. We know that LL37 breaches the membranes. We know that it causes oxidative stress, but we don't really know when they happen in relation to one another. And that knowing that order will actually help us to figure out exactly what this peptide is doing. So now we have both of the dyes, Cytox and a different red dye whose name I can't remember, but basically the green burst is not Cytox, sorry, the cell rocks. So when we see green, that's the oxidative burst, and then when we see red, that's the inner membrane breach. So we know that they're temporally correlated. Doing it in two stains in the same bacteria can allow us to really get a solid handle as to what the order of these events happening is. Okay, so at 0, we add the peptide. Then, okay, we see that green burst. And then right after, we get that red burst. So it looks like it takes a little bit of time for that oxidative stress to start. But relatively soon after that, we get that inner membrane breach, which is very cool. It's very interesting how that's very tight in terms of temporal correlation. That's very soon one after another. And so when you plot the data into a graph, you actually see that that temporal correlation is very close to one another, right? So this is fluorescence intensity. This is the time lapse. And so RHLL37, that's actually the peptide that has had a fluorescent group added to it. Welcome more juice. We've got graphs. We're talking about how this particular antimicrobial peptide LL37 destroys cells. And we'll see that we have a hypothesis that it causes oxidative stress, and indeed, the oxidative stress peak, it peaks just before the membrane breach happens, just before, as indicated by this green part. So we stay low the entire time. At about 16 minutes, we get that green spike first, and then we get that red spike right after. So that's really, really cool to see that that's so tightly correlated.
Yeah, I don't know if you have the other graph, because there's like, I'm looking at the paper right now. I don't know if you are, but there's two graphs that look near identical that talk about different ones, because they both use red fluorescence. The other dye is actually Cytox orange, which is very confusing now. There's cell rocks green, Cytox green, and other Cytox orange. And Cytox orange is what they use to measure the permeabilization of the membrane. And they're just using orange because it's a different color, because they can't use Cytox green because cell rocks is already green.
Yeah.
But then the one you had showed regarding RHLL37, which I don't know what the RH stands for.
I think
Red
it's
something.
Rho. I think no, Rho is a
Um,
protein. It can't be Rho. I don't know. I didn't read that
Oh,
carefully
here,
to the
I just
methods.
look. I just like control F through the paper. Rhodamine? I don't
Rhodamine.
know.
Oh yeah,
I
yeah.
can't.
Rhodamine is a red dye. Rhodamine is a red dye. Yeah,
Oh,
yeah, yeah.
okay. I did not
Wow,
know.
dude, I pulled that from my grad school years. I was like, Rho is a protein, but rhodamine is a dye. Yeah, yeah, yeah.
And they said it wasn't going to be useful for anything.
Yeah,
But yeah,
permeable. Oh,
the
sorry. Yeah.
one regarding RHLO37 is also showing the accumulation of the peptide inside the cell as the membrane is becoming permeabilized, and as it's attacking it, more and more can keep accumulating in the cell, causing it to fluoresce a bit more.
Yeah, I actually cut that for time. Sorry.
Oh, sorry. Well, let me just extend it by bringing it up anyways.
No worries. Permeabilize is a wonderfully cromulent word. Indeed, it
What
is.
is cromulent?
No need to worry too much.
Yes.
That's why I actually replaced. You weren't here for the beginning, but permeabilization, I just changed it to membrane busting because it sounds cool.
Yeah.
It's more badass. So as a recap, actually, Morjuice, you joined really appropriately at the recap point, so that if you didn't pop in for the rest of the stream, here's a good point to jump back in.
Mm-hmm.
So this is what we learned about this antimicrobial peptide. We learned that it can poke holes in both the outer and inner membrane, do this membrane bust. And we also know that it causes oxidative stress. And we also know that LL37 decreases bacterial growth. So this is what we've learned so far, right?
Mm-hmm.
So one would imagine that in between the permeabilization of the outer and the inner membrane, that this is where LL37 must cause oxidative stress, right? And so I'm gonna be a little coy. I'm gonna be a little coy here, right? Because here's another one of these logic puzzles that I'm about to drop on you. Is there a control that you would like to see? I just made an assumption. My assumption was LL37 causes oxidative stress. And that's a big assumption to make here.
Mm-hmm.
And I want to give chat a little bit of time. You don't need to have seen the rest of the presentation. Just these statements alone can prompt you to thinking about whether or not there's a control experiment missing. Certainly, when we add LL37, oxidative stress happens, but does LL37 itself cause oxidative stress? That's kind of interesting, right? This is kind of an interesting question because all of these things are correlated. We add LL37 and we see that the membrane busts. So I'm pretty confident, because that's the first thing that happens, that LL37 genuinely pokes holes in the outer membrane. But what about the rest? Because of the timing, we know that LL37 likely permeabilizes the outer membrane. But after that, I don't think it's very clear what LL37 is doing. Not sure what oxidative stress is. Oh, so basically your body is always trying to destroy itself, and what's stopping you from destroying yourself is things like blueberries that soak up the stress molecules that come out of your mitochondria. That's like my 101 5-minute version. When you see antioxidants in your blueberries, it's that.
Yes, oxygen good because it's reactive, but sometimes oxygen bad because it's too reactive and it'll just destroy stuff. So whenever there's too much destructive oxygen, it's oxidative stress.
Doing the experiment in anaerobic environment, we will get to that. So you have predicted a future and past experiment. Phi is really pushing the blueberries today. It's my official sponsor from the FDA. Do we happen to know if LL37 works on a gram-positive? That's a question for Spollax. I don't really know if that's true.
I don't know.
But I do see Thomas the thermonuclear bomb on the right path, so we will continue. What happens when the membrane is busted with something else? That's actually more than the right path. That's exactly the control that we need. So very good, Thomas.
According to a cursory search, apparently LL37 is active against both gram-positive and gram-negative. Although I would be interested into looking into how it actually works against gram-positives, because I'm only really familiar with gram. I work pretty exclusively with gram-negative bacteria, so that'd be cool to see how it has activity against both.
Yeah. Life on Earth is, in a way, literally on fire all the time. That's true. Your mitochondria
Yes.
are combustion reactors where they are constantly making tiny little fires.
Hmm.
Very cool. Okay, but yeah, but props to Thomas. You get a gold star. That's exactly the experiment that we need to see.
Can I?
Unfortunately, oh yeah, sorry. What's up?
Oh, I was like, can I just briefly talk about gram-positive, negative?
Oh, sure, sure.
Yes, gram-positive and gram-negative bacteria are just two very broad classes of bacteria that kind of describe their cell cell wall slash cell membrane, the outer layer. So a gram-positive, the name comes from gram staining. It's just a type of stain that is used, and they noticed that whenever they use the stain, there are certain bacteria that would stain a certain way, so they call that gram positive, and then the ones that didn't, what they called gram negative. Gram positives have two major layers, where it's just they have a really, really thick outer layer of the certain protein called protein molecule called peptidoglycan, and then they have an inner membrane beneath that big thick layer. And gram negative has like a peptidoglycan sandwich where it has an outer membrane, a little thin layer of peptidoglycan in the middle, and then a inner membrane. It's just the broad way to describe two different classes of bacteria.
Cool. Thank you for that.
Yes.
So let's go off of Thomas the thermonuclear bombs idea. What happens when you use something that isn't LL37? Well, the authors actually did this in a previous paper that, because it's not open access, I can't actually show you their data, but I'll just summarize it here. So a previous paper that they did, it's the same lab, they used soap. So there are detergents that can bust membranes. If you add just enough of it, it'll bust only the outer one and not the inner one. So you titrate it. You basically make the correct concentration of soap to just dissolve that outer membrane. And what they saw was that when you do that, you don't generate that oxidative stress. So that is fantastic. That is exactly the experiment that we need to do before making all these assumptions. No access through your school's equivalent account. No, I can see it. I can't show you because YouTube's running ads on this. How am I going to get my $50 worth of ad revenue if I show copyright material?
Am I gonna get a half of that $50 for being here?
Yeah, man. If you want like if you want to pull gotcha at like one of those what is it, the gumball machines, I'll let you
Yeah.
I'll let you pull a gumball machine.
I'm expecting one gumball in payment, or a handful of like those little banana candies.
Exactly. Although arguable. I don't think you could pay me a quarter to eat them. But yeah, good job, Thomas. That's exactly the control. The answer is yes. LL37 must be doing something else because the soap experiment does not generate oxidative stress, but LL37 does. So this is their hypothesis. They want to get granular. Again, they want as much mechanistic information as possible.
Oh. Sorry, I just don't like this diagram.
No, I'm sorry. I know I couldn't. I was trying to find something that was, not going to get me in trouble with advertisers. So I picked the mitochondria.
Fair.
But LL37 is hypothesized to disrupt the electron transport chain. So for those of you who don't know, inside your mitochondria is a bunch of proteins that live in the inner membrane that shuttle electrons around and use those electrons to generate electromotive force for an enzyme called ATP synthase. That enzyme, that pump, it's kind of like a salt pepper grinder that grinds electrons. Yeah, that uses electrons to spin to push ADP and A and phosphate together to generate ATP, which is the cellular fuel that your cell uses.
Mm-hmm.
And so I know this is mitochondria and not your bacteria, so this is not what it looks like in E. coli, but for the life of me, I could not find a picture that I could use, so just bear with me. In any case, the machinery is somewhat similar, and this is actually what, I forget who had it earlier in the chat. I think Alexis was spoiling this.
Yes, yeah, she will.
Yeah, Alexis was spoiling this. Bro, this diagram is straight out of the AP Bio textbook. Well, I think it's copyright Creative Commons license 4.0.
Nobody tattle. This will be our little secret.
Yeah, bro, the Krebs cycle. Yeah, exactly. But the idea here is that one way that oxidative stress can be generated is through disruption of the electron transport chain. So this complicated thing that happens in the mitochondria that allows you to generate a thick load of that's a bad way to say it, a dummy like thick amount of ATP.
Where say so.
Where say so, exactly. Say so channel, say so cell. the thing that allows you to generate that dummy thick amount of ATP from oxygen are these complexes, these protein complexes that live in these membranes. But the thing is we can test whether or not LL37 needs that machinery by using cyanide. We already
That's
have
fun.
membrane busting, and why not thick load? God. God.
What have we done?
Oh,
What
no.
have you done?
What have I done? I should have just not addressed it and pretended like I didn't know what I said.
Yeah.
That would have been the say-so move, actually. So
Moving
actually,
on.
Yeah, moving on. Cyanide. So cyanide is a very interesting molecule, of course, very deadly, very famously deadly, but not many people know why it's deadly. Cyanide basically causes you to effectively drown on the inside in the way that it binds to the iron in the electron carrier cytochrome BO3BD. That's the cytochrome in bacteria. It binds some other cytochrome in eukaryotes. And basically, it stops O2 from moving down the pathway. So you need to shuttle oxygen, or you need to pass electrons to oxygen to make it into water, as we can see in this complex 4. So I don't actually know Spollax. If you have Google ready, can you check if cyanide inhibits complex 4? Because I don't want to spread misinformation. Almonds, yes. So for those of you who don't know, cyanide smells like almonds. But it basically binds to the proteins of your mitochondria and causes you to be unable to generate energy. And if you can't generate energy, that's not good. Yeah, I think it binds.
It does.
Complex 4.
I saw two different things where cyanide is traditionally viewed as a cytotoxic agent, with its primary mode of action being the inhibition of mitochondrial complex number 4. But then there's just another paper right underneath that said cyanide did not affect the expression of complex 4, but I'm sure they're just like up or down regulating something or else.
I think it blocks something or another, from what I remember how cyanide works, although that could also be misinformation. Anyway, cyanide...
It inhibits complex 4. You were correct on that.
Great. Wow, dude. Graduate school, yo. They're gonna be so proud that I remembered a thing about cyanide. Yeah, so let's check whether or not using cyanide to block this process affects whether or not LL37 is capable of producing oxidative stress. So this is a monster of a bar graph, so let's go through it one by one. So we're using cell rocks. This measures how stressed your cells are oxidatively. So how much superoxide your cells are making. And so if you don't add any, bacteria, E. coli, are actually quite capable of handling the generation of reactive oxygen species. So we don't get a lot of oxidative stress. This is just your average, normal E. coli going about its day, not really worrying too much.
Mm-hmm.
Let's add that peptide LL37, and all of a sudden, bang, you get that huge burst of oxidative stress. At the maximum point, that's a lot of oxidative stress. But now let's check whether or not cyanide, which blocks the electron transport chain, which is one way that we can do oxidative stress, affects LL37. So before we get into that, what is your hypothesis, chat? I'm covering this bar graph with a white box, keeping it away from your eyes.
Mm-hmm.
But if we hypothesize that LL37 does in fact cause oxidative stress through the electron transport chain, and we block the electron transport chain, what's the size of that next graph, that next bar? What's that size? I think there are probably other... I think a couple of the chatters had mentioned earlier a lot of interesting other ideas that I would like to scroll up and revisit to see whether or not that would address the same question or similar. Oh no, their hypothesis is very specific to the proteins here. Smaller than the second bar, small. You are both correct. Very, very good, very good. So you add that cyanide, and you get a massive reduction back to wild type, no LL37. So you go from extremely stressed to just as stressed, because this difference is not statistically significant. It's basically the same. It's almost as if you didn't add any peptide at all with regards to oxidative stress. So adding cyanide completely stops LL37 from working, which is very cool. So this is the conclusion. LL37 causes oxidative stress through the inhibition of the electron transport chain because the addition of cyanide, which blocks the ETC, stops LL37 from working. Is the structure of LL37 related to the cyanide? That's a very interesting hypothesis. Cyanide is a very small molecule. It's basically Oh god, it's the nitrogen attached to a carbon. Spollax, can you help?
Yeah, it's just like carbon attached to nitrogen. I don't know what the bond is, but it's a teeny tiny molecule,
Yeah, teeny
a
tiny.
molecule, if you will. Yes.
Oh, a small molecule. That's awesome. But LL37 is a small peptide. Compared to cyanide, it's much larger and differently structured. Does this mean that LL37 could be used to counteract the effect of cyanide as a poison? That's not a bad hypothesis. However, there's not enough information here right now to. So, for example, we don't know whether or not LL37 binds to the same cytochrome. We just know that adding cyanide stops it from working. So that's not a bad hypothesis. In fact, if More Juice's hypothesis is correct, how could you test that? That's kind of interesting. If More Juice's idea about using LL37 to stop cyanide poisoning, if that was true, how could you test that? Hello, Chad. Welcome.
Yes.
Oh, no, I'm asking chat in general, but also,
Okay.
yeah, Spollax, I'd like to pick your brain, put you on the spot.
Sure. Sorry, I'm also getting hung up on the fact where it's like, hypothetically, if it did work, then it might just kill you / the bacteria through LL37 instead of cyanide. So it's kind of like a pick your poison kind of scenario. But because cyanide disrupts the electron transport chain and disrupts the formation of reactive oxygen species, you could hopefully use one of the cell roxes, or maybe it's just cell rox green, to hopefully determine the presence of those reactive oxygen species after introduction of LL37. But that's a very rough.
Yeah, I would say that my answer would be a lot more simpler than yours.
Yeah.
I would just dump actually, never mind. My experiment doesn't work. What I was going to say is that I would add a lot more LL37 and see if it kicks the cyanide off the cytochrome. But that doesn't work because if you add a ton of LL37, you just kill the bacteria. You can only do that in purified format. So if I took just cytochrome BAC plus cyanide, and then I could try to add LL37 until it rips that cyanide off. But that's not very informative when you do it in a dish like that.
Yeah, in vitro, there's a little bit of
hand waviness
magic that happens. Yeah, where you can't really apply it too much, but
it's not a bad idea. It's just not as good as I want it to be.
I did like that idea.
Yeah,
but protein purification is hard, especially membrane proteins. But we don't have to talk about that.
Oh yeah, maybe for another day.
Yeah.
I would like to do the lactose intolerance, dude. Do like the lactose intolerance and tread on myself.
Do not consume cyanide under any circumstances.
Do not ingest. Do not
any amount of cyanide.
Oh no, not the hypothetical lactose intolerance guy. Yes, very hypothetical. If you don't know what I'm talking about, chat, I have a stream where I dug into a very totally hypothetical, not a real YouTuber that I'll get in trouble with, what their self-do-it-yourself gene therapy experiment was like. You can check that in my VODs. Okay, so there's an issue with this, okay? There's one small issue here, and as a scientist, I cannot help but pick up on all of these annoying little issues. Is that this is a completely reasonable conclusion to make, but is there anything potentially wrong with concluding this conclusion on the left where LL37 causes oxidative stress through inhibition of ETC because we added cyanide which blocks LL37? Anything potentially wrong? Can we draw this conclusion, or how confident are we that what I'm saying is correct? That LL37 stops electron transport chain because we use cyanide. I'll let chat think about it for a little longer while I address Chad's comment. For legal reasons, for legal reasons, I can neither confirm nor deny that is the YouTuber that I am talking about. But yeah, that puts bag of almonds down. Yeah, well, you know. I don't remember how many almonds you would have to eat to get this effect to happen.
I think it's like the amount of just like the sheer volume would kill you rather than the cyanide.
Yeah,
it's
like
that thing where
your membrane rupture.
If you eat like 3 million bananas, you'll die of radiation poisoning, but you also probably die first of eating 3 million bananas.
All right, it's getting a little late into the presentation, so I'll maybe get this one out of the way. This is another correlation thing, right? LL37's reduction of oxidative stress is correlated with the addition of cyanide. Ah, no, super annoying. So if we want to get around this idea that this is correlative, is there something that you could do to get a little more confident that this is correct? And I'll let chat chew on that for a little bit. I know it's getting a little late into the stream, so the brain might be fizzing out. But yeah, like this might be annoying for certain people, but for basic scientists, I love like when I catch myself going, "Oh, but is this really causation?" Like that's the most like actually thing ever.
That's really what science is. You just actually yourself and your peers and everyone.
In grad school, you learn how to actually yourself all the time. It's actually one of the best benefits of going to grad school.
Mm-hmm.
Both have the same active site. Like that's one hypothesis, yeah. But I'm trying to see whether or not there's an issue with concluding that it affects the electron transport chain because you use cyanide.
Mm-hmm.
Cyanide is the problematic part here. The usage of cyanide.
Oh, hints.
So, Spollax, do you have an answer for me?
Not a good one prepared. My brain is starting to leak a little bit out of my shell.
Oh, actually, more juice. Even though you're in weekend mode, you actually got it. Can you use something else besides cyanide to get the same effect? Yes.
Ooh.
Very good, very good. Just to eliminate the interaction between LL37 and cyanide. Exactly. Through this interacting web of correlation, causation, variables coming in and out, it's really hard to conclude that this is because it blocks ETC and not because cyanide doing something funny. So if we get something else that does the exact same thing as cyanide, but it's not cyanide, that would give us more confidence. It wouldn't like give us 100% confidence, but it would definitely give us a lot more confidence that we're right. And that's exactly what they did. Maybe cyanide is doing something completely different that happens to protect the cell from oxidative stress just by coincidence.
Mm-hmm.
And so using a different molecule like CCCP, another electron transport chain disruptor, it also works in the exact same way, providing more evidence that LL37 acts on the electron transport chain. Very good.
I'm sorry, I'm in the way of the figure.
Oh, yeah, I can.
It does say CCCP.
Yeah, but behind Spollax right now. To be fair, I think Alexis asked a similar question earlier, so I'll just take a quarter of the credit. Thank you very much for sharing that credit. Very nice.
Mm-hmm.
And I'll acknowledge Alexis as well. I might not have read that answer very properly. Okay. So this is, I believe, what Morjuice had also recommended, doing this entire thing under anaerobic conditions. And what we see is what we would expect, that if we don't have oxygen in there, then we don't get oxidative stress. That's kind of to be expected. What's kind of interesting is that when we pump up the LL37, so this entire thing, this left-hand side was done in 4 micromolar. If you increase the LLC37 fourfold, you don't really see that big of an increase. In fact, it's not statistically different than not having any at all. It's really blocked out very heavily by not having oxygen around. Very cool. Yeah. So this is where we were before this experiment. This order of battle permeabilizes the outer membrane, which is correlated with oxidative stress, which is correlated with permeabilization of the inner membrane, which is correlated with bacterial growth being inhibited. Now we can actually put a link here. LLC permeabilizes the outer membrane, and it causes oxidative stress through the generation of superoxides through the ETC. So this is their, we'll call it, what is it called? Schematic, an overview schematic. So this is how the authors believe that this peptide kills bacteria. It busts through the outer membrane, and then it lodges itself in the inner membrane, this cytoplasmic membrane. This causes some pores to open, which, you know, is bad for the cell. But also, it might be doing something to block the transfer. Like, it blocks this enzyme somehow. And instead of using oxygen to generate water, we're using it to generate superoxides. And what the peptide is doing is it's shifting the balance between generating water and generating this molecule that destroys you. Very cool. So this has been sort of interesting on a mechanistic point of view. I always find molecular mechanism to be interesting. This is kind of my bread and butter in terms of like what I like to read.
Mm.
Oh, Alexis asks if it has affinity to cholesterol. That's actually not cholesterol. This is something called the NDH complex, which I could not tell you more about. It's one of these electron carriers that shifts electrons into different places and eventually ends up as water. But yeah, superoxides sound cool, so they're probably bad for me, right? Yeah.
Unfortunately.
Most of the cool things in biology are terrible for you.
Yeah.
So again, so I find anything to do with how molecules work inside a cell to be interesting. Just knowing that just inherently is cool. But what makes this result cool on like a treatment level, if you want to think more about real people and less abstractly, this implies something that the oxygen environment that a cell lives in can influence how effective the peptide is. So you can imagine that different tissues in the body have different levels of oxygenation. Could this logic be applied to other antimicrobial peptides? So maybe LL37 is better at attacking bacteria that live in places in your body that have access to oxygen. So maybe LL37 could be good for something like treating a wound, right? An infected wound, like bacteria accumulating on the surface of a wound, some place where they get that oxygen. And maybe it's not very good for targeting those bacteria that live in anaerobic portions of, say, your gut. It's very interesting. H2O2, a superoxide, close as a peroxide. Yep, nice. Yeah. Although that is, I do believe H2O2 does count as a reactive oxygen species. So it is in the same family of superoxides.
I think they mentioned a couple times where I think it might be cell rocks also react similarly to H2O2. They also qualify a couple times. I think that it doesn't like react to everything at all possible.
Yeah, they were really thorough about things I didn't care about.
Yeah, that's another part of like what was like I was getting hung up on it was just like how they were qualifying a lot of their experiments. So I was like, okay, and then I was still getting just like hung up on all these different little things that they're talking about.
Yeah, reading papers is quite difficult, which is why I do the hard work of framing it, and then you can do the fun work of thinking about it. But yeah, I think this is more or less the end. Yep, that's the end of the slides. So good job, Chad. I think like I'm really having a ton of fun with these journal clubs because what I'm learning is that my chat is sometimes smarter than me. Like I went to Spollax and I was like, I don't know why this bacteria bust open, but it did. And then I think some of you had figured it out very quickly. Y'all are actually so smart. It's very impressive.
This is not an easy paper, nor an easy subject, nor like is it presented well. Credit where credit's due. Fi did an incredible job of making this digestible in this presentation.
Thank you.
Hats off to him. If I had a hat and hands, I would take my hat off, but I cannot. But y'all are also very smart.
Yeah, probably from blood. That would be the answer to your question, Hair. They get their own circulation from the circulatory system. Take the top shell off, says Surprise Pants. I don't know that we can.
I think that would kill me.
It would also not be safe for YouTube. Say so, stream.
Yes.
But yeah, I hope. Like, I don't know. I think it's pretty fun. I want to know. I'll do it as a poll. Whoa. Yeah, no, we are say so. We all say so. Promise. Trust. Do you like the aspect of getting asked questions? Oh no, don't plug your only clams. Don't plug your only clams.
We were say so. I'm kidding.
Today marks the end of say so. So do you like the process of getting asked these sort of questions? Because I know that in school I hated this kind of thing because I was just like, just tell me so I can write it down so I can study for my tests.
So I can go home and take a nap so I can go wake up to study again.
Exactly. So I can memorize all the steps in glycolysis. I don't need to be challenged.
The Krebs cycle.
Oh no, Phi the Lutrophil. That would be up for Toxo because I have no control over whether or not Phi the Lutrophil happens. That's all Toxo. Love getting to exercise my brain and experimental design skills.
Yeah. These are very, very useful skills to have.
Yeah, I think they're just kind of fun. Like, even if you never do science, even if you never do science, just disambiguating between correlation and causation on a regular basis, I find I don't know about you, Spollax, but I feel like knowing how to ask questions as to how to arrive at the truth has made me like a more balanced person.
Yeah, it really. How do I want to phrase this?
Like I
Um,
feel like I know myself better because I'm able to ask myself questions about myself in a more
yeah,
rigorous way.
yeah. It definitely applies like a level of introspection. It also applies, there's a good amount of like skepticism that's healthy, I think, just towards like information that you pass by in your day-to-day life, and this kind of thinking, for lack of a better term, like a scientist, is useful.
Yeah,
It can also be it can also be very fun.
it's also not to like blow my head up or anything, but like but like actually like the skill is very useful to be able to disambiguate correlation causation, but I have to acknowledge that like it doesn't toxic can tell you in our day-to-day lives living together that that doesn't mean that I'm smart. Like
Yeah.
the skill of critical thinking is just like woodworking or learning how to paint, right? It's just something that you can turn on when you need, but it doesn't
Hmm.
make you like a higher IQ person. It just makes you better at this very specific thing, which is investigating the truth. Um,
Yeah.
and that's not necessarily easy if you go outside of the science realm, right? Like truth in the social context is more A, philosophy, B, journalism. But if you're talking about observing natural phenomenon and trying to figure out how that natural phenomenon is connected, I think this exercise is a lot of fun.
Hmm.
Critical thinking and being smart, that's correlation, not causation. Nice. Yeah, let's
Yeah,
go.
exactly. You get
Let's
it.
go.
More juice gets us.
Yeah. That's awesome. All right. So that's kind of it. Spolex, do you have any like final? I'm not gonna kick you out like right now.
Sure. Yeah.
But if we want to just like wrap up what we thought about the paper and like from your experience, I figured it'd be kind of cool to give you the soapbox of where this kind of research tends to go, because this is my first antimicrobial peptide paper. Um,
Yeah.
it's a very new world to me.
So I can just briefly like broadly talk about just like research papers, I guess in general, because I mentioned this to Fi while we were just chatting right before stream started, is that there's kind of like a couple of different categories of research paper in my mind. Some of them are there to learn about a brand new topic or to, explore a new subject, or I mean, sometimes they're just papers that are just there to be entertaining. Sometimes you can find them entertaining in a way. And then there's also papers sometimes that you use as like a resource. And like what I mean by that is that like for something like this, with this kind of information dense, very specific paper, I wouldn't be like reading this kind of normally unless I really wanted to learn specifically maybe about like these kind of assays. Like I could see myself in like the workplace in my lab using this to just maybe study up on like a Cytox green or a Cellrox green or Cytox orange assay or something to see how I could apply it. But this kind of research, there's a lot of this kind of like this is like a mechanist. It's a more of a mechanistic exploration of like these potential new antimicrobials or these new molecules, and then something you can do after this is kind of taking that one step further by engineering different characteristics that you would like an antimicrobial peptide, for example, to have. So now that you figured out that it does this, or it's a natural sequence, or it does this, what can we tweak to make it better or worse, or target a different thing? And then in doing so, you kind of can understand it more. And maybe after one research paper you wouldn't be able to, but after the cumulative effect of all these labs studying all these different aspects, eventually you can learn how to basically just like build and design these kinds of new antimicrobials. And then that's where like big changes can be made. It's kind of the cumulative effect of sometimes someone just does a mechanistic exploration first, and then finally somewhere down the line people can actually start using that base knowledge to create their own magical protein that will solve all the problems in the world.
Very
There
neat.
there is literally my boss slash my PI was literally just talking about this, how one new drug had, maybe it was just not the fact, maybe it wasn't a new antimicrobial peptide, but that there's a new class of antibiotics which have actually been made it either through FDA drug trials or like are in the process of and pretty far drug trials. But there are different antimicrobial peptides that exist. They come with caveats, but so does everything.
Yeah. I want to address
Oh,
the results of the poll that I had put up with 12 votes is unanimous yes, which is
100%. Wow, look at that.
that's just awesome. Actually, I can
Yeah.
use that data in the grant applications that I'll be applying for. Because I wanna go big, y'all. It's like real talk. Like my ambitions for this channel are like sky high. Like I'm the type. Toxic can confirm this too. I'm the type of person where if I have a dream, I don't go halfway. I will
Mm.
work if I believe in myself. I will work like really hard to achieve. And so the fact that I have unanimous, well, it's 12, so it's not a huge like N, but to show a grant agency that like there is this audience of people who enjoy the stream format where it's kind of like a lecture where you can just jump in at any time and participate and have access to somebody who can actually ask you the questions back. The fact that the reception of, and actually, if I talk about grant application and applying for the grant, if I take a look at my what is it, CCV, concurrent viewers, I can actually take a ratio of the people that said yes to my concurrent viewer count, which is usually about 20-ish. And if I can say that half the people who watch me really enjoy the interactive component of this new way of learning, that could be very compelling for a grant agency, because if the ratio is half and I ask them, hey, give me money so I can do self-promo, so I can pay artists, then just imagine that if I had like a much larger audience, if half of those people are interested, that's incredible. That's an incredible get. Whether or not there's actually an audience for me out there, I don't really know. But I'm very encouraged and heartened by the fact that it's not just me. Because here's the thing, when I do this in class, the students do not like it.
Yeah.
The students do not like getting grilled like this, but I'm like, I make sure to not be very mean. I'm like, just come up with an answer, throw an idea at the wall. Getting your brain to do something is better than idling. This is how you become better, is just trying. So we're getting a cut of that grant money, right?
Yeah, is this gonna be like that big RO1 grant money? You're gonna be bringing in
RO1. A little now. Who's putting expectations on who?
a little ambitious.
You'll get a cut of the grant money abstractly. Maybe I'll gift subs if actually, if I get the grant, I will gift subs. I'll gift like if I get the one that I'm thinking of, I could gift a lot of subs. That would be really fun to do.
An RO1 grant, just to explain it, is like the big boy. This the big dog grant. It's like the ones that big labs use for like multi-year projects, and it's millions of dollars brought into a lab. They're very difficult to get because it is a lot of money.
They're also like, if you go for NIH, their grants are in excess of 100 pages that you have to write. It's ridiculous.
Yeah,
it's really big boy stuff.
Yeah, my boss will just like disappear, not for weeks on end, but like days of the week. He'll just be like, I'm writing. Okay, bye.
Yeah.
And then he'll just be plugging away writing grants.
You know what you could do? Let us show us up on your lectures. Actually, that's not a bad idea. If I have to submit proof of principle, it might be. Oh, I'm gonna have to ask chat on that day to be on their best behavior. No talking about.
Yeah.
No pointing out that I said membrane busting.
No only clams. No
okay.
Thick ATP or anything like that.
No thick loads.
Yeah. Oh, Alexis also. I saw earlier. You were talking about carbonpentamases, and that would be great. Maybe I should do something about that.
Yeah,
let's do your Thunderbin. I'll just talk about carbonpentamases over on Twitch. If you're interested in doing another journal club, this has been a lot of fun. Even
yeah, I'm really glad.
Very close. I feel like honestly, like I was like kind of kicking myself. I was like, man, I kind of wish I worked on more of these antimicrobial peptides in my lab so that I could actually be kind of the expert on this. But carbonpentamases are a little bit closer too. Big fan.
I would love.
No, not a fan of them because they cause antibiotics and bacteria, but I think they're interesting.
Oh no. Okay, so what you're saying, Chad, is I shouldn't warn you when I'm going to be appealing for a grant. I should not let chat know ahead of time, because this is not I am not getting confidence.
Can you imagine if your committee is just watching your stream?
I mean, actually, on Cideray, who's like my VTheorem co-streamer,
Hmm.
we're part of the same group. She's had people from her professional background tune into her streams before, and I'm like, I have been very careful to be very anonymous
Yes,
because I'm very careful with my privacy.
I also am as well. I had one very close friend who's also like works in my lab. The streams have crossed very slightly, but I'm still being very cautious about it.
Mhm. Yeah, I don't know. I think that's a good question, more Juice. How does Sai not cringe at that thought? I think that if you are in her field, you might encounter fellow weirdos at a higher rate than my
Mhm.
field. That girl be built different. Agreed.
Weirdos in a very complimentary sense of the word.
Yes, yes, yes. As
But
much
I
love
understand
as I
exactly what you mean.
called Sykin a weirdo, or no, I called Sykin a freak. That's too far. No, no, no. I think she hangs out with weirdos, but Sykin's a straight up freak. Weirdos, parentheses, complimentary. Exactly, exactly. But I say that with all the affection in the world for Sykin as well. So Sykin MD was a doctor that I had, a medical doctor, and I called him a freak because doctors work too hard. Look, all VTubers are kind of weirdos. Yeah, yeah, exactly.
Yeah.
Agreed.
We're all different. I mean, most people are a different flavor of weird. Or some flavor of weird, but it's just about finding the complementary kind of weirdo so that you can talk about antimicrobial peptides for an entire stream.
Exactly. I'm weird, but I'm your weirdo. Yeah.
Exactly.
Yeah, so I mean, if you want to talk about antimicrobial peptides, I mean, you kind of know what my streaming schedule is for the rest of the summer in terms of like pretty booked out for a while, but come the school year, when I get back into my normal groove and not like full time making content like a madman, that would be fun actually.
Yeah,
that'd be really cool. I would love to collab with other VTubers on their specific science stuff because I have such a good time learning.
I am bacteria boy and also lab management boy.
Yeah, I mean that's also very interesting life too. I was so jealous of all the labs that actually had a lab manager. So your work is definitely appreciated, at least in the abstract, by me. Because let me tell you, it's not fun doing all of the work for everything that you specifically have to do.
Yeah, I try not to let it go to my head, but it's nice to feel useful, I guess. I'm trying to be a positive force for the world and also in my lab.
Oh yeah, more juice. So on the lab horror stories idea, so Spollax and Resta actually did a stream where they talked about which lab reagents and machinery they feared the most. It was a lot of fun.
Yeah, it was a lab of fear tier list.
Yeah, I loved that. I thought that was such a fun stream. It's such a good idea. I was chatting up a storm in your chat too.
Yeah, that was a lot of fun. I had a whale of a time with that stream.
I think like where I felt very related to you in terms of like like people don't like if you're in a lab you don't want to look like a weakling to your like fellow grad students, so you don't talk about what you are afraid of in the lab. Like,
yeah.
So they'd be like, yeah, I'm afraid of the liquid nitrogen too, or like I'm really afraid of the centrifuge. Like,
yeah, oh my god, the centrifuge. It's a very humanizing experience in lab, I think, to come to terms with what you're afraid of and also just things that you don't like.
Yeah,
things you're not super stoked to work about, especially like in a professional environment. Maybe it's not the best to be like, I don't like working with this, but recreationally, I'd be like, man, I do not like Arabidopsis models or plant models.
Yeah, some of them are just. I have lab experiences so bad I actually don't want to share them on stream, not out of embarrassment or anything, but because they're really sad.
Oh.
What's with the centrifuge? So Thomas, a centrifuge is a machine that spins samples very quickly to sediment things by their density. The thing that makes them scary is their size. The bigger the centrifuge is, the more potential damage you can do with it. In fact, in every safety training I've ever been, like all the required videos that you have to watch before you operate a centrifuge, they're always like, if you unbalance a centrifuge, it can become a ballistic missile, and it killed these people. And I was like, whoa, that's crazy. And so the way that they train you to handle the centrifuge is that if it makes a bad noise, then you shut it off immediately. What they don't show you is what that sound sounds like. So every time you run the centrifuge, you have to sit there and you have to question yourself and go, is this a normal centrifuge noise or am I about to die?
Yeah, it's. I haven't been around, but I have heard a couple of centrifuge failures, and they are shockingly loud. It's kind of one of those things where like you'll probably know whenever it happens. But I also like that typically the standard operating procedure for if a centrifuge is about to go ballistic is to shut it off or unplug it, and after that the next step is to run away as fast as possible or put as many things as you can in between you and the centrifuge so that in case it does take off, the potential damage is minimized. The oldest reactive I have in the lab. Fortunately, my lab is pretty new. We didn't, and also since we're a biology lab, we don't tend to work with a lot of super dangerous chemicals. I think every day that I am not a chemist because that's where the really sketchy and scary stuff is. Um, we inherited maybe some stuff from like the early 2000s from another lab, but I will say I was recently, I mean, earlier this week, I was working with a type of media that is from 2006, and it expired in 2010, and it still works. It is just very, very hard because it absorbs so much moisture out of the air.
Yeah.
It's a very pretty green color though. If I had a picture, I could. I do have pictures, but it's too difficult to show. I think right now.
That would be really neat. So it's actually not the RPM that matters. RPM is just how fast it spins. It's actually how much G it can generate because G can vary based on the rotor radius. Basically, it spins with a lot of force, and the higher that force is, the more dangerous it is. It sounds like a plane. It does sound like a plane, but when it ramps up, especially the larger centrifuges, when it ramps up, it can be kind of scary. It's like a roller coaster. I've actually had to stop multiple undergrads from from being the victim of a ballistic collision because undergrads do not always remember to balance their centrifuges. And let me tell you, I've never sprinted so fast in my and I give myself a lot of crap for not. I don't believe I am a better person than anybody else, right? Like I don't believe I have an inherent morality to me that makes me more ethical or more moral than any other person. But when I learned how to cut myself some slack, when I heard that centrifuge start running off balance, my body, I didn't even think, my body threw my body between the student and the centrifuge to turn the centrifuge off. Like I didn't, if I had actually thought about it, maybe I would have run away.
Yeah.
But like the like reactive heroism, like, you know, again, I'm not saying I'm a better person. I'm saying in a split-second decision, my body chose to put my body between me and the students. Which was actually kind of scary for me because I'm like, I should probably prioritize my own safety, just like realistically. But that's not what ended up happening.
I'm more valuable than a freshman.
Yeah, exactly. What's a freshman doing? I don't know. Do I put dummy samples in a centrifuge to balance it? Yeah, you have to. If you load 10 mils on one end, you just take 10 mils of water on the other end, or buffer if you want to be super nerdy about it.
We call them blanks. So I was talking about it, I guess, in my stream. I can briefly talk about it now. Is that we have different sizes of centrifuge. So we have like a small benchtop centrifuge, which spins like a mill's worth of liquid at a time, like one milliliter. And then we have a tabletop centrifuge, which spins up to like 50 milliliters at a time. And then we have a floor centrifuge, which spins up to 400 milliliters at a time. And the floor centrifuge is the one I'm the most afraid of because there's so much force involved in that one, and there's so much mass that it's moving around at such a high speed that that one could go up to like 30,000 Gs or like 17,000 times the force of gravity, which is very liable to go through walls if it's unbalanced. And the way we do that is that whenever we have to use blanks or just even putting anything in it, you have to measure things out to within 1 gram of each other for like standard operating procedure. An old grad student told me that if hey, there's a picture of my Bunsen burner. I just sent a picture of the plates to Phi. If it's within 1 gram of each other and the machine breaks, it's not your fault. If it's outside of 1 gram of each other and the machine breaks, then it is your fault. But yeah, so these are the medias I was talking about. So the green colored one, which I took a picture because I very rarely get to work with such colorful media. It's usually just like a really boring pale yellow color. But the green
RGB petri dish.
it's stoplight colors.
That's very funny.
Rest had mentioned that earlier. The green media is called TCBS, which I have to double check what it actually is. Stands for thiosulfate citrate bile salts sucrose agar. It's a selective auger that only grows Vibrio cholera and other Vibrio related bacteria. So any of the other ones won't actually grow on that plate. So we were using it to select for a specific type of bacteria. The red one is called McConkey auger, and it's got like a lot of fun dyes in it, whatnot. And it's selective for gram-negative bacteria, the one we were talking about earlier with like the peptidoglycan sandwich. It
Spollax, you're cutting out, I believe.
just a regular LB,
Yeah.
which is like the most generic media we have in lab, along with an antibiotic called rifampicin, which is like this very dark red orange color. So even when I put a little bit amount of it, it'll actually turn the plate slightly orange. Oh, did I disappear? Oh, there I am.
Yeah, I think you're back. You mentioned Vibrio. I would love to talk about Vibrio, right? Because they are the biofilm. What is it? Quorum sensing bacteria, right? Do you do quorum sensing with them, or?
We don't do quorum sensing. That was something that my boss actually was like, "Hey, do you want to try doing this?" But we ended up not pursuing that project. Um, I've done a decent amount of work with Vibrio. Not a crazy amount. Unfortunately, nothing in regards to quorum sensing or biofilm. I think someone in my lab has done biofilm stuff with Vibrio before though. But Vibrio is an interesting pathogen and also an interesting bacteria to work with.
This is why I'm like every time I finish an idea for content during the process, we'll talk about something, and then like 3 more content ideas will pop up. It's like this is why I'm like, oh, at this rate I will never finish all of I'll never finish biology.
It's a good problem to have.
Yeah.
Bobtail squid bacteria?
I
Vibrio?
don't
It could
know.
be. Yeah, Vibrio is it's very commonly found just like in waterways, waterborne pathogen. So I guess it wouldn't be too crazy. Um, one one thing that I realized earlier was that the bacteria that causes botulism, tetanus, and like C. diff, they're all in the same genus, Clostridium botulinum, difficile, and tetanium or something like that. I don't remember what it is, but that's quite the group, like the mean group of bacteria, I guess.
The
Yeah,
squad
the
pulls
evil
up.
squad.
All
Vibrio
right.
fischeri? I don't. Maybe I'm not familiar with that strain.
I think Fisher is the one I was referring to, the
Oh, okay.
biofilm model.
Oh, tetani. There you go. Thank you. And cyanobacteria for like biofilm work or what other kind of works? Oh, Parahemolyticus. Yeah, that's also a real annoying strain. Annoying in the sense that it's like a human pathogen.
Yeah, it might be. Yeah, so on my channel, I tend to do molecular mechanism stuff. But
Hmm.
if we were to collab again, I don't want to plan too far ahead because I can't guarantee you in the. I Spollax, I got myself real deep into setting up content for the summer. I got real deep into planning content, and I still,
Big
of course,
dreams.
yeah,
You should
exactly.
try not to burn out. Do
But
your best. Do your best.
if we were to collab again, it might be
Oh,
more fun to have you pick one to discuss, to pick a
sure.
paper to discuss. Because I kind of gave
I would
Spollax
love
3
to.
options, and I tried to keep it molecular because that's like my channel. But
Hmm.
it'd be cool to kind of just like chat because everything on my channel. I was telling the side about this earlier. Is like I'm like my energy is very organized, right? I have a presentation. I'm very focused, and I get through my presentation in a lot amount of time. But it would be fun to just kind of shoot the shit about, whatever bacteria. Because even though I'm a mammal eukaryote person, I like
Oh
love
man,
learning about
eukaryotes.
bacteria.
You can. I have a lot of basically like I mean I think all science is cool. Um, I just have like different
Okay,
beef, I guess, with
we'll end
certain
it on the
types.
beef. We'll end it on the beef because we talked a
Oh
little
sure.
bit about beef. So let's get into this beef. Okay, so what is your deal, man,
With what?
with eukaryotes?
The eukaryotes, they're just difficult. Eukaryotes are hard. Eukaryotes have so many different like they're difficult to culture. They grow slower. Like working with mammalian cells is kind of a pain. Bacteria, like they grow really fast. They're kind of they're kind of sloppy in that way. I think they're a lot cheaper to work with at times. But I think the biggest thing is that they're just like I'm already having enough difficulty understanding prokaryotes at times, and eukaryotes just seem so much more complex to me. Um, and that's why I'm just like, oh man, like I have a ton of respect for all the people who do like, mammalian cell work and immunology work and whatnot. But I'm like, I will just happily stay over here in my bacteria world where my culture grows in a day, and it costs me about 6 cents to maybe to make. And so the risk of me messing something up and ruining an experiment is pretty low.
I think it's so interesting because I kind of have that attitude about plants. Because like
Oh,
for me,
that's
bacteria, eukaryotes, yeah, bacteria and eukaryotes I'm fine with. Like fungi, they're a little weird. And I know
hmm
that plants technically count as eukaryotes too. But I think what strikes me is that I can handle like the difference between bacteria and eukaryotes kind of in my head because of the interest factor, right? Like I'm interested in how human beings work from my days as a pre-med, and I like bacteria because they're more simple, which means you can understand the molecules a bit better. The story
hmm
isn't as complicated. Yeah, I mean you're right. Like eukaryotes have a whole bunch of crap going on, and bacteria are more simple. Although that doesn't mean that they are simple. It just that means
Yeah.
by definition they have fewer things inside their genome, so there are fewer things you have to study. But when I learned about plants, I was like, I just finished learning the Calvin cycle in AP Bio. I just finished learning the mitochondria. And then like 2 days later, there's the Calvin cycle in photosynthesis. And I'm like, that makes no sense.
Yeah.
The fact that it's split up in two phases is also just mind-boggling. So I think the Calvin cycle ruined plants for me. Um,
Plants are just like there's kind of like not a universal like way that life is understood, but I guess for the sake of this right now with our beef with plants is that kind of like a through line with all these living things, and then plants come along and ruin everything with being haploid and diploid and
yeah,
tetraploid and
yeah, yeah.
just like throwing this like the life cycle doesn't make sense and they grow weird and there's too many of them and there's too many different classes and I'm like I don't like this.
Yeah, and they have what alteration of generation. I think that's when I put my foot down. I'm like, oh my god, I can't believe plants have like two sexual reproductive life cycles. Like,
I
this is
I,
too much.
I have so much respect for plant biologists. The work they do is super important. Plants are really cool. I hate Arabidopsis. I've been to so many talks about Arabidopsis, and it makes me want to bang my head against the wall
But
sometimes.
and again, shout out to my fellow V Theorem member.
Yes.
Nepenthes VT. She's a lovely, lovely horticulturalist, loves plants. But I straight up told her, I was like, dude, I got beef with plants, man. And then they were like, well, I got beef with animals, and I'm like, or not all animals, where
Yeah.
they like bugs, but yeah, like okay, so I'm glad we kind of I'm glad I figured out or I learned the origin of your beef. It is not
Yes.
that you find eukaryotes
It's
to
not.
be.
Yeah. No. It's not like I hate eukaryote scientists or anything like that. It's just that I find them very difficult to work with. And that honestly, that also maybe be a fun collab. It's just everyone talking about their least favorite subjects or areas of interest or fields or whatnot, things they dislike in their own field.
Oh yeah,
Hold on.
I
There's a
would.
couple.
Yeah. What's up?
Sorry, no, there's just a couple of things in chat I wanted to address. Where one
Mhm.
of them was like how to culture like cyanobacteria. I don't know that one specifically. I do know that there actually is a lot of work going into like being able to culture different like wild strains of bacteria because things like E. coli and whatnot they're very easy. But there's definitely a lot of ones where especially where like brand new species that have that's kind of untreaded ground. Yeah. Where they actually do put a lot of look into like what specific nutrients do they need, what kind of conditions do they need. Sometimes it's possible, sometimes it's not possible. There are some bacteria that they just can't for whatever reason culture in a laboratory environment. And also Serratia marcescens is a very cool bacteria. It's kind of found everywhere, and this is a fun fact. Y'all can do science at home. If you have in your bathroom like your shower or your bathtub or your sink, if you see any like pink mold, quote unquote, that's not actually mold. It is Serratia marcescens. It's a type of bacteria. It likes to grow in those kind of wet, humid, and damp environments. It is not that dangerous unless you are immunocompromised or you have like large open wounds. I wouldn't go rubbing up against those pink stains, but yeah, if you ever see any like pink stains in the bathrooms, I know they're very common in like college dormitory showers and whatnot, like public baths. That's Serratia marcescens. Fun fact.
I think Serratia is also one of the biofilm model organisms as well because that pink slime is a biofilm. So
Yeah,
that's
I think it would make sense. I don't know for certain, so I can't say with
Yeah,
with
I
all the
think
gumption, but
yeah.
it would make sense given where they live, and also like I've stepped on it in my shower and it's like a little slippery. I'm like that's probably not good. I need to clean my shower.
You're outing
Um,
yourself. My
I,
bathroom
I
is
clean it
pinkless.
I clean it regularly, but they just installed my bathtub in a really poor way where it pools water whenever I'm done taking a shower. It's the apartment's fault, not mine.
Yeah.
But another fun fact, I guess, about Serratia is that the way they first discovered it was in Italy. They had, because it's kind of a ubiquitous bacteria, it's kind of present in, like the soil and the environment everywhere. They left out a bowl of polenta on accident, and it ended up spoiling, and it had this red coloration, which is very indicative of Serratia. And they called it like bleeding polenta because it had turned red or looked like there's like blood on top of it. But when they sampled it, it was actually a bacteria that produces a very distinct pigment. Serratia's cool.
Yeah, it's pretty sick. I mean, maybe we'll collab about Serratia. I'm pretty
We
interested
could,
too.
yeah.
All right. It's getting late on my end. Thank you so much, Spollax,
Of
for
course.
joining me and getting to geek out about bacteria. Thank you all
Yeah.
for joining and really providing some very excellent uh responses. Are all like, I don't know. Maybe I had too low expectations of stream chat, but y'all are like, you are on it, and I'm very happy about what was objectively like a very hard paper for two professionals to read. You were able to
Hmm.
follow along logically with the experiment, which is very, very exciting. Um,
Yeah. Thank you so much, Fi, for having me, for putting the work together to get this presentation. Uh, yeah, I don't want to pressure you or anything, especially now that it seems like your schedule is quite busy, but I
oh
had a
no,
whale of a time, so I would be more than willing to come back here and be grilled
literally
live
all
on
of my
stream.
content is biology, so of course I'm very happy. I guess I'll end it on the sweet note, on like a very sincere thank you to Spollax
Aw.
for being vulnerable and coming on to show his whole ass on what he does or doesn't know about bacteria. Because I talk about biology for a living. My actual job is biology, right? I mean, Spollax's job is biology too, but his job is to
Hmm.
do biology. My job is to
Yeah.
read and talk about biology. And it's not very easy to just because you're a scientist doesn't mean that you are good at talking about science or even comfortable about talking about science in front of a live audience. In fact, if you see that, there are a lot of scientist VTubers out there, but I don't think a lot of them are that comfortable with talking about science online. So I was very happy to learn that Spollax, because I reached out like very soon after learning that he had even a little bit, like the second I learned you were a biologist, I was like, I sent a DM like
Immediately.
the following day being like, do you want to talk about biology? You don't have to. I told them that he didn't have to read the paper. I was just like, just please come on stream, you don't have to read a paper. Like, I just want somebody to talk to. um so yeah, so just as a very sincere thank you to Spollax for bearing his
Oh,
whole ass out here
thank you, thank you.
and really providing me with good company while we talk about this paper and offering your own unique insights and your own
Hmm.
expertise, which is very valuable because I'm not an actual microbiologist. I'm an amateur. I'm an appreciator. I'm a professional molecular biologist, but an amateur microbiologist. So I was very happy to have a professional come here with me and talk. And there'll be more of this stuff in the future. I've got a couple of additional collabs that have to be finalized, but yeah, it was a lot of fun. Until next time, Spollax, just make sure that Spollax's socials are in the description of this video. So if you want to go follow him on
Yes.
Twitter and Twitch. He plays, I think, a lot of fighting games, right? That's what I've
Oh,
seen.
not yeah, not. I've been trying to get back into it, but just like a lot of games in general.
Mhm.
Unfortunately, that's the benefit and the curse of being a variety streamer is that whenever someone's like, "What do you do?" and I'm like, "I don't know, just video games, I guess." So if y'all are interested in that,
All right,
but
cool. With that, bye, y'all. Bye
take care,
bye.
y'all. Woohoo! This is a fun end screen. I like it. That was great. I mean, like maybe around like the like 2/3 mark. Whenever I was like, I feel my brain leaking out of my shell. I'm just like getting tired after like I'm just like trying to think again, but I could tell that like I just wasn't able to focus on the paper super well. Which I mean that happens, especially because today was a little bit of a busy day for me. But I know I can work around it. I still have